Hello, everyone. My name is Bahtiyar Yilmaz, and I'm joining to this meeting from the University of Bern in Switzerland. Today, I would like to share our latest study, which is about understanding the plasticity of the adult intestinal microbiota. In our previous study in 2019, we showed that the IBD microbiota is different than the non-IBD patient, but in between the Crohn's disease and the colitis patient, also there are huge differences. As you can see, many of the bacteria in the Crohn's disease patients are reduced compared to the colitis. And this is already shown by many groups. So, but what we found, what was important in our study, that we show the patient who had the surgery has a completely different microbiota than the no surgery patient. And this is also represented on the right side in this panel in the bar chart. You can see the patient who never goes under the surgery, they have a lot of bacteria that are related to the short chain fatty acid producing higher in their gut, where those bacteria are reduced in the surgery patient. And we know that almost 20-30% of the patients who have the surgery, then they can relapse and they can have the active disease again. And we think that that's because of not having these bacteria in their gut is the main reason. But we never show actually if this is really true. So we never show whether the patient who went to the surgery actually before the surgery, if they have a different microbiota. What is this? We never show what is the effect of the surgery on the microbiota itself. So based on this, and another finding that I'm not showing in here actually is where we see that the purgative is actually homogenizing the gut, and then we don't see any difference between the ileum and the colon. So hence we wanted to actually characterize the microbiota in ileum and the colon patient where there is no purgative. And for that, we use the stoma patient and we try to answer to the following questions. So we wanted to understand how much the surgery affect the microbiota. Is it really disrupting, or the microbiota is already in a dysbiosis situation before the surgery? And what is the really true ileum and colon microbiota differences in the patient? Because the ileum is a different compartment to access without the purging. So, and for that, as I said, for that reason, we use the stoma patient, and we wanted to see what is the true microbiota in the ileum samples. And lastly, we wanted to characterize the fasting and the feeding effect in the ileal microbiota. So that's one of the things that is never characterized, since, as I said, that ileum is not an easy component to have an access. So for that, we use the three different patient groups. So in the first one, where we have the patient before the surgery, we got some content samples from the rectum and then the biopsies. And at the time of the surgery, we got also ileal rectum and the rectal content and the biopsy samples. And we follow the patient after the second surgery, where the stoma is closed and the patient goes completely normal to their life. And the second group is, we call them the cured patients. So those are still, they have the stoma back, but they're completely healthy. So because the cancer area or the inflamed tissue is removed, and in that reason that they are completely healthy, but still they have the stoma, and that's giving us an advantage to get the ileostoma right and left colostoma sample easily. And in the last group, as I said, we just used 6 patients where we fast them overnight, and in the morning after the first sampling, we provide them the breakfast, and then we sample every 2 hours, and in the lunchtime we provide we provide the lunch actually, and then we follow them again up to 10 hours during the day. So from the first part of the study, where we have the surgery, as I said, we are trying to understand the effect of the surgery, and what we see is that at the time of the surgery, the ileostoma microbiota is relatively better. After the surgery, is a bit higher. There is a reduction after the surgery, but is not significant in ileum, and this is also true for the rectum part of the gut. And in the beta diversity, there is no significant difference in the ileum, so meaning that the ileum microbiota is not really affected from the surgery, but the rectal part is affected because the colon is kind of disconnected, and the rectal is just feeding from the mucus shedding actually. And for that reason, during those sampling time, the microbiota change a bit, but after the surgery, it reverts back. But one of the important things that we see in this study is actually the E. coli is blooming, as you can see on the bottom part of the slide, so that the patient that they have a relatively high level of the E. coli from the beginning at the time of the surgery, but that's also blooming a bit higher after the surgery, and in some patients this stays even higher after the closing, even after they have the second surgery. So in the second part of the study, as I said, we wanted to see what is the true microbiota in the ileal compartment. So since it's so difficult to enter without without the purging. So in this case, we took advantage of this Ileum, ileostoma, and the colostoma samples, and then we can have a direct access to the sample without any purging. And when we compare, we see that the ileal microbiota, as we expecting, is lower than the colon left and right compartment, and it's lower than the feces for this richness. And as we expect, the proteobacteria are higher in the ileum compared to the colon, and this is also true for the IBD. But as we know that in IBD, the proteobacteria are even higher in the ileum compared to the colorectal cancer patient. And one quick note, so for those who wants to study with this type of samples in the future, so you don't need really this to go directly in the intestine. You can even use the stoma bag. We compare the stoma bag content from the inside of the stoma itself, from the intestine itself. There is no difference between the microbiota from these two type of samples, and there is no metabolically also difference. So you have a quite similar profile in both of those. And the last part of the study, that's the most kind of interesting part also, to see how much is the fasting is affecting and the feeding is affecting the microbiota during the day. So this one was our setup. As I said, every 2 hours we sample and we provide the food in the breakfast and the lunch. And then we check also metabolically, which I'm not showing here, but you can, those who are interested, they can look at our paper. But then in here, we do the shotgun metagenomics, and we try to see what type of substrains are appearing over a time based on the feeding. To our surprise, so we see so much host DNA from the beginning, so which was quite interesting, because these are the contents of where you will not expect so much the host DNA, but that's the reality. And at the time of the zero, after the overnight fasting, what we see actually 99 or 90% of the DNAs are belongs to the host rather than the microbial. But as soon as we give the food, we provide the food, so then that's the microbial DNA is increasing, and during the days, as you see, for each patient is fluctuating, and every time after providing the meal, the fast-growing bacteria are increasing. And we also quantify this with instead of the relative numbers. So we had the bacterial count, and what we see in after 2 hours, it goes 1 or 2 logs almost in some patients, the bacterial count per gram of feces. And the richness is fluctuating a bit, but in reality, is actually it's reducing a bit because of the increase of E. coli and Klebsiella, those type of fast-growing bacteria after the feeding. This is the overall taxonomy profile. What we see, the most abundant bacteria in these patients are the E. coli and the Klebsiella, actually. So, and then those are after they eat or during the day, it's relatively growing in a fast manner. But like, as I said, our interest was about to see, okay, so providing the food, is it giving a different advantage to some of the substrains to grow better? And if you just focus on the right side to do this yellow color, so that's a patient, one of the patient, initially in the first sample is almost 100% similar to the database, but as soon as we provide the food, so genetic, as you see, it's reduced to the 98% similarity. This is like we measure actually the population annual nucleotide identity, meaning that it's like almost 2% of the genome of this bacteria is different, meaning that the different substrains appear. And if you compare between the patient, as you see, every patient has an E. coli, but they have a different type of, or different substrains of the E. coli, or different subspecies of the E. coli. And one last thing, that, okay, so we can see that there are a lot of variations, so that there are a lot of changes during the day based on the fasting and feeding status. But we also look at the structural variants in the E. coli, or in the paper also you can find the Klebsiella, and what we see that it's not only this specific SNPs are appearing, or the specific substrain with the different SNPs are appearing during the day, but we can see also there are a lot of structural variants are appearing, whether they are deletion or the variable structural variants. So, and many of them are related with the machinery of the bacteria to adapt to the environment. So, such as like the membrane and the transporter and the phage related. And without entering the detail, I just want to conclude our study. So we show that the biomass in the lower intestine substantially shed in the ileostoma after the feeding, and the colostoma microbiota or the colon microbiota is relatively stable, but there are minor changes during the day while they are healing. And there is a very rapid substrains appearance and disappearance based on what they feed. And I think that's really important, because that's the reason why maybe many of the bacteria are not appearing from our gut, even in the catastrophic events. because there are some substrain that there who are professional to be adapted in this environment and help to survive of that strain specifically in our gut. And this study will never be possible without many clinicians and then the Swiss IBD cohort and our collaborator. I would like to thank to all of them and all the funding agency. And I will be happy to have your questions if you have any. Thank you very much.